Welcome to the intermediary metabolism class. Now let us discuss HMP shunt pathway. So HMP shunt is one of the major important pathways whose precursor molecule is glucose. From glucose will be producing different pentose sugars, especially the ribose. And ribose will be utilized for different purposes. So we'll see that in the significance of HMP shunt. Apart from this pentose sugar, we also produce reducing equivalents. So those reducing equivalents will be used for the synthetic processes, especially the fatty acid synthesis, as well as the synthesis of DNA and RNA structures, the nucleotides. So let us start with the HMP shunt pathway. So the first precursor will be the glucose, and glucose 6-phosphate will undergo dehydrogenation reaction with the help of glucose 6-phosphate dehydrogenase enzyme, where we produce one reducing equivalent, which is NADPH plus H+, which will lead to the production of 6-phosphogluconate delta lactone. 6-phosphogluconate delta lactone, by means of lactonase enzyme, we convert it to 6-phosphogluconate, where we introduce water molecule hydrolyze the lactone ring structure, and we get 6-phosphogluconate. 6-phosphogluconate undergo further dehydrogenation reaction, where we produce NADPH, which is the second reducing equivalent in the production of in the HMP shunt pathway, where we get ribulose 5-phosphate. From the ribulose 5-phosphate, if isomerase is acting upon, of course, ribulose 5-phosphate will be converted to ribose 5-phosphate, and if epimerase is acting upon, ribulose 5-phosphate will be converted to xylulose 5-phosphate. Now, this ribulose 5-phosphate and ribose 5-phosphate combines with the help of transketolase enzyme to produce cedoheptulose 7-phosphate, a 7-carbon compound, and a 3-carbon compound glyceraldehyde 3-phosphate. And again, the 7-carbon compound and the 3-carbon compound will combine to form a 6-carbon compound and a 4-carbon compound. The 6-carbon compound, which is fructose 6-phosphate, and a 4-carbon compound, which is erythrose 4-phosphate, by means of trans-aldolase enzyme. Again, with the help of trans-ketolase enzyme, a 4-carbon compound, erythrose 4-phosphate, will combine with a 5-carbon compound, xylulose 5-phosphate, to form a 6-carbon fructose 6-phosphate and a 3-carbon glyceraldehyde 3-phosphate. So this will be the final carbohydrate which is formed with the help of your HMP shunt. So glucose 6-phosphate will be converted to different hexo sugar, especially the monosaccharides, as well as the pentose sugars, and those pentose sugars, example the ribose 5-phosphate, will be utilized for the nucleotide synthesis. And the two reducing equivalents which are produced from reaction 1 and reaction 3 will be used for different purposes in the body. So this we will study in the significance of the HMP shunt. Let us discuss about the significance of the HMP shunt. Already we have discussed it gives enormous amount of a ribose sugar as well as the reducing equivalent. The reducing equivalent, which is produced by means of HMP shunt pathway, will be used to scavenge the free radicals which are produced by means of different reactions, cell reactions, especially the immunological reaction, or the oxygen concentration if it proceeds or if it exceeds in the cell than the normal. So these free radicals will be scavenged by means of these reducing equivalents. So in case if it is not scavenging these free radicals will destroy DNA, proteins, fatty acids, and all the biomolecular structure, which leads to the complete destruction of the cell. And apart from that, the reducing equivalents also is used to maintain the membrane integrity of your RBC cells. So RBC cell membrane integrity is maintained with the help of NADPH. Apart from that, it also prevents the formation of methylhemoglobin, and also it is used in a number of detoxification reactions which is present in the liver enzymes. So those enzymes require the NADPH reducing equivalent in order to detoxify the drugs whichever it is entering into the body, and also the toxins which is entering into the body. So both the detoxification will be performed with the help of the reducing equivalent NADPH, which is produced by means of HMP shunt. And also the enormous availability of ribose sugar. The ribose sugar will be used majorly for the synthesis of RNA and DNA, and also for the synthesis of coenzymes, where the nucleotide is the essential component in the structures. So these are the major functions or significance of the HMP shunt pathway.